Hello everyone. In this video lecture we are going to discuss about bacterial cell, gram staining and gram staining technique. So without any delay let's start our today's topic. So here it is shown that bacterial cell. Under electron microscope we can see the structure of bacterial cell. They are having RNA, glycosomes, lipid globules, mesosome, cell wall, respiratory enzymes, pilus, filament, hook, flagella, mucilage, plasma membrane, nucleoids, ribosomes, mucilage, and you can all see from this diagram. So bacterial cell is a prokaryotic cell because they are only having one cell and they don't have a defined nucleus. They don't have a proper nuclear membrane around their nucleus. So their genetic material is called as nucleolus or nucleoid. So moving moving toward the gram staining technique, we need to discuss about what is gram staining. So what is gram staining? Gram staining is developed by the scientist Gram. That's why it's known as gram staining, and it is a very special technique in which we use to classify bacteria into two groups like gram positive bacteria and gram negative bacteria. Those bacteria that take up the gram stain are called gram positive, and the other one that do not take up the stain, that is known as gram negative bacteria. So now let's discuss about the gram staining technique. So what we are doing first we prepare a bacterial smear. So what we do now, we have to take a glass slide on which with the help of inoculating loop we can take up some colonies of bacteria from the agar plate. Then we just put that on the glass slide and we spread with the help of other glass slide by making 45-degree angle, and we can just spread that smear on the glass slide. After this we have to air dry it. After few minutes when it dries, we put some crystal violet stain on it, few drops, and we have just left it for 30 second. Wait for 30 second. After 30 second the bacterial get stained with bluish purple color. Then what we have to do, we just wash with the water gently. We have to wash with the water, not very. Then what we have to do, then we just put some iodine staining, like 0.5% solution of iodine, which is act as a moderant. Moderant means it helps in the making formation of the crystal violet iodine complex, so that the cell of bacteria get properly stained and absorb all the stains of crystal violet. Then after this, we wash the iodine solution. Then we put some alcohol. When we add alcohol on the bacterial smear, what we see, we have two different cases. For example, some bacteria, first case, some bacteria retain the stain, so that they are called as gram positive bacteria, because we can see that color is purple like. But some bacteria destained, that the whole color of blue color or purple color is washed off after adding alcohol. So and that in that case we have to counter stain with safranin. We put some solution of the safranin on the bacteria, and after that we wash the safranin solution, and we see that pink color appearance. So the if the color of the bacteria is pink color, that means they are gram negative, and if the color is blue, that means they are gram positive. So by the help of this technique we can distinguish between the gram positive bacteria and the gram negative bacteria. So why the gram positive bacteria are bluish in color and they take up the stain? Because they are having a layer of peptidoglycan, which is a very thick layer which absorbs the blue color. But in case of gram negative, this opposite, it has a very thin layer of peptidoglycan. That's why when we treat with alcohol, the whole color, purple color, is washed off. And by adding counter stain saffron, pink color, it appears like a pink color. So thank you. Hope it will helpful for you. Subscribe for more such videos. Okay, bye.